And I will have pleasure to lead this session. Now we have 5 presentations to listen. And without further delay, I would like to invite Marcin Tomasz Mazurkiewicz, who presents us the impact of forest management and climate change on biophyte diversity on the close to natural forests. The floor is yours.
Okay. So thank you very much for this privilege to open this session. I think it's a privilege. And so I'm Marcin Mazurkiewicz. I'm from Białowieża Geobotanical Station of University of Warsaw, and I would like to talk to you about my PhD project. And I'm working on impact of forest management and climate change on bryophyte diversity in Białowieża forest. So first things first, where is Białowieża forest? It's a forest complex on the border between Poland and Belarus. I work on the Polish side, and we have 2 areas, 2 sites. The first one is located in strict reserve of Białowieża National Park, right here, and it is an area around 140 hectares and divided to squares 100 per 100 meters. And we have very similar area also divided in those squares in the managed forest in Białowieża. And so I work only in this managed part, and because this part from protected forest data were collected in 2015, and so we have this data, and I currently working here. This will be only preliminary results because I'm on my first year, and there's a lot to do. But what data I collect? So I do an inventarization of bryophytes in these squares, and I collect, of course, presence of each bryophyte species, also their frequency in scale from 1 to 3, from rare to common, and also I note in which terrain microforms they occur, on which from 24 plus 1 to the other substrates they are, and we have also only 6 types of forest, and this is a table from Project Crypto, which was done in 1992 by Professor Faliński and his co-workers. And like I said, we have only 6 types of forest, and the most common is subcontinental oak hornbeam forest. And in this table, you can see what part of those areas have, for example, from this oak hornbeam forest, the 35% of this area in protected forest was this oak hornbeam, and in my managed area only 34. The differences are not so big, so we can say that this forest from protected and from managed forest are quite similar, and we have a couple of hypotheses. I will focus on this first 2. They are about differences between taxonomic and functional diversity between those two areas. The third and the fourth hypothesis need first more data and more time to do some analysis, so there will be only those, those two first. So, going to results. Here is a PCA ordination of bryophyte species composition. What we can see here is each dot is 1 square, and blue dots are from managed forest, green dots are from protected forest. And we can see that according to species composition, those squares from managed forests are first very different from this from protected, and the second are very homogeneous. I will show you the next slide also about number of species. Those forests in managed part of Białowieża forest are very similar. On the other hand, the protected part is much more divergent. Okay, so about some parameters of this taxonomical diversity. Here we have a box plot with species richness, and we can see that the species richness in protected forest is much more is much higher than in managed. And this the same is with Shannon index of biodiversity. It is higher in protected forest, and what we can see also that these managed squares have very similar number of species, and in this protected there were bigger differences between them. So about the hypothesis number 2, for this functional diversity, we used calculated parameters of functional diversity, and I will explain what does this mean. And we also used bryophyte traits, which were calculated by Van Zuyen last year. And here I will show you those couple of bryophyte traits per square. So about these parameters, we have 4 of them. Here are 2. There is functional divergence and functional evenness. I need to add that we don't have the same number of squares from managed and from protected forest, so this can change in the future, I hope. Because as we can see, and statistically and on this box plot, we can see that there are no differences, or these differences are not so big between them. And those parameters, to be honest, for me are still quite complicated. So functional divergence is about proportion of abundance with the most extreme values of bryophyte traits, and functional evenness are measuring the regularity of the distribution of species in this niche space. Here we have also 2 more parameters, which is functional dispersion and functional richness. And here we have some differences, especially in functional richness. About functional dispersion, statistically, in Wilcoxon test, there were differences. On the box plots also we can see something. So, as I said, I hope that in the future there will be this, at least it will stay like that. About those bryophyte traits, And here are box plots with community-weighted means of some of those traits. They're calculated per squares. And then we wanted to find if there were some differences between protected and managed forests, and of course they were. So about spore size, it's still a discussion if it's valuable or important trait. The bigger are spores, well, on the other hand, the smaller are spores of bryophytes, the longer distance they can travel to disperse the species. This is probably because in managed forests we have less species and more generalist species. So like that. And about moisture and light and temperature. Wait. About moisture, we see that in protected forests there are species that prefer more wet substrata, which will be incorrect because in strict reserve there are more, for example, dead wood. And from continentality, light, acidity, and temperature, we see that in managed forests we have more species that like the higher values of these traits. So I see that my time is over. So if you will have any question, we have discussion. And if this is not enough, don't be afraid to contact me on this email. And also it is in our book abstracts. Thank you.
Thank you very much for your interesting talk. Actually, you have 5 minutes left. However, still we will have the discussion. We have half an hour discussion after the whole presentation. So now I would like to invite Francesco Laterini, who will who give us a talk about evaluating small-scale harvesting disturbance to the forest soil in Mediterranean beach high forests. The floor is yours.
Thank you very much for the introduction, and brief introduction mine about the framework of in which I carried out this, let's say, this research, which is my Marie Curie grant that I'm currently carrying out at the Polish Academy of Sciences. It's a research project of 2 years in which I am studying the effect of active forest management on European beech forest in Italy and Poland. I'm going to define a bit better later what I mean with active forest management. So, first of all, in these specific cases, I will present some results of my overall project, specifically focused on small-scale forestry. What I mean with small-scale forestry. In the context of central and southern Italy, forest operations are generally carried out by low or medium forest enterprises, generally family enterprises, which are not using dedicated forest machinery like skidders and forwarders, but are generally relying on agricultural tractor adapted somehow to the forest work. So, little enterprises, few workers, and machines which are not specifically developed for the forest work. The research hypotheses that we had while establishing this experimental design were mostly 3. So, first of all, the first hypothesis is that the impact, the disturbance to the forest soil related to the, let's say, the harvesting intervention in beech forest in this area carried out in the framework of small-scale forestry is related only to the mechanization applied. So, we have an impact to the soil only in the soil affected by the passage of the machine, while instead in the soil not affected by the passage of the machine, the impact is negligible. Third hypothesis is that there is a strict correlation between a disturbance to soil physicochemical properties and the disturbance to soil microarthropods biodiversity. This is the experimental design which I'm applying in practically all my research. And I think it will help to better define the hypothesis. So, in any harvesting subcompartment, we have two portions of soils. In orange, you can see the so-called undisturbed or low disturbance soil, in which the intervention was carried out, but the machine didn't pass. While instead, the blue lines are the skid trails network, and it is the soil materially affected by the passage of the tractor, the skidder, or the forwarder. To compare, let's say, to understand the impacts related to the silvicultural treatment that were applied and the harvesting system that we are using, we have to compare with an unharvested control area, in green in the image. All along my project, I took into consideration 5 study areas, 2 in Poland and 3 in Italy. But today, as I told you, I will do a focus on one of the study areas in central Italy. In this study area, located in the Abruzzo region Apennine, I identified 6 forest parcels. 3 control, so 3 unharvested parcel, and per every control, very close to this control area, there was a parcel, a subcompartment, harvested 1 year before the surveying, so in 2021. So, we have 6 parcels, 3 harvested and 3 unharvested. And within the harvested ones, we have the disturbed soil, obviously, and the undisturbed soil. To let you understand, very beautiful this scheme. This is, let's say, the harvesting system which is applied there. Short wood system, so 1 meter long logs, extracted by an agricultural tractor equipped with these bins, which are called forwarding bins. So, the material is not directly touching the ground like during the usual skidding operations in beech forest, but however, with this system, the operator is forced to do many passages on the same skid trails to extract the same amount of timber as for skidding. The parameters that we evaluated were related to soil compaction, so soil bulk density, penetration resistance, and shear resistance, the soil organic material, and the soil microarthropod community evaluated by a qualitative index called QBSR. Fast to the results. So, 3 experimental treatments. Ecco, a posto. Imagine. Yes? Break? Okay. It was protest. So, the result. 3 experimental treatments. Control. Disturbed soil and undisturbed soil. As you can see, apart from bulk density, for all the investigated parameters, there is a statistically significant difference only in the disturbed soil. As expected, there is more compaction, so higher penetration resistance and shear resistance, and there is lower soil organic material, and most importantly, lower QBSR index. So less biodiversity of soil microarthropods in the soil sampled in the skid trails. While instead, in the undisturbed treatment, there is practically no difference in comparison to the control area. To answer the third hypothesis, we did also models linking the soil physicochemical parameters with the microarthropod biodiversity. As we can see, also in this case, bulk density didn't respond very well, but the other 3 variables, penetration resistance, shear resistance, and soil organic material, were statistically correlated with the soil microarthropods community. Indeed, higher values of compaction and lower values of soil organic material means less biodiversity of soil microarthropods. The RDA analysis is practically confirming this. As we can see, the QBS high values of biological soil microarthropods and soil organic material are related with the control and undisturbed treatments, while high value of soil compaction are related with the disturbed soil. So the research hypotheses were confirmed, and what we have to explain why. So we demonstrated that the silvicultural treatment, so in this case thinning, with a biomass removal ranging from 20 to 25% of the overall biomass in the stand, didn't cause a significant alteration in the soil, both from the point of view of compaction and from the point of view of biodiversity of soil microarthropods. This is probably related to the fact that the canopy cover in thinning interventions is practically negligible if it is done in the correct way. So the amount of light and of rainfall which is reaching the soil is comparable to the one in an unharvested forest. On the other hand, we have a strong, significant impact on the ski trails, even if the machine used is much lighter in comparison to a skidder or a forwarder. In comparison to a forwarder, we are speaking about almost a 10 times less heavy machine. Here, about 4 or 5 tons, a forwarder loaded can arrive to 40-50 tons. But the impact is the same. Why? Because with this system, to extract the same amount of timber, you have to go, I don't know, 30-40 times, going back on the same trail. So this is also the importance of planning the forest interventions. So also, when we listen to the fact that we have to apply small-scale harvesting because it's less impact than normal forest management, it's not all the time like this. Also, when we are applying small-scale harvesting and little machines, the so-called reduced impact logging harvesting systems, also in this case, we have to plan the interventions. We have to try to avoid the strong soil compaction. So all those best management practices which are generally recommended for forwarders or skidders application, like doing the soil trafficability map before the intervention or placing the residues on the trails to decrease the compaction, should be applied also in this case. Or even better, maybe try to change, but I know that it can be complicated with the local loggers. Another possible solution that I'm strongly recommending in steep slopes terrain like this, because in those parcels the slope was about 30-35%, after obviously a training, after knowing how to use the system, is much better to shift to aerial extraction by cable yarding, because in comparison to ground-based machinery, these systems are really not impactful on the soil, because simply there is no contact of the log with the ground. And I think that it is everything. I thank you all for the attention, and stop.
Thank you very much for your talk. Because we have parallel session, it would be better to stick with time. So now we have time for one or two questions. So maybe somebody would like to ask about.
No?
Okay, so if not, please keep it in mind, or maybe note somewhere, and we will return to this presentation. So now I would like to invite Crispin Ilunga Mulala to give us a talk about performances of native tree species in plantation, a synthesis of the Guineo-Congolian region.
Thank you very much. Now we will travel to tropical forests. We'll talk about the performances of native tree species in plantations, a synthesis for the Guineo-Congolian region. A global change affects countries around the world differently, and each country tries to make the project to mitigate this global challenge this global problem. And as you can see in this map, the risk of many tree species is high in the tropical area. And in the context of tropical Africa, we can see many degraded areas. And then the degraded area is can be the result of change in land use, maybe for agriculture, from logging, and we can find many degraded area in that space. And many forest stakeholders think that planting can be one of the solutions to mitigate the problem of global change. And several initiatives has been launched in recent years. We can give the example of IFA 100. And the target depended on each country. But what can we learn from previous results in reforestation in tropical Africa? We can note that since colonial times, planting trial has been carried out, but those results are scattered, and we do not have a global view of tree performances in the plantation. And in the context of this study, we asked the question, what can we learn from previous research in the whole Guineo-Congolian region in Africa? This is an example of the trees planted since colonial times. And as you can see on this photo, we have good performances of trees. What was done to have those kind of performances? Yeah, sorry. Then we synthesized the survival and growth performance of native tree species planted in the whole Guineo-Congolian region from Africa. And tested whether the performances metric depended on environmental conditions, planting methods, and species traits. We did a meta-analysis, and we select 45 studies, which gave us, which was distributed around 38 seats in the whole Guineo-Congolian region. 89 tree species was in our data set, and provide 687 observations for pioneer species, non-pioneer, light-demanding, and shade-bearer species. As you can find in our observations, we have many pioneer and non-pioneer light-demanding species. Then we ran linear mixed effect models. Here is the example of the model on the diameter growth. And we did the model for true survival, high, and diameter growth. And in the fixed effect, we consider the planting age, the planting methods, and we found in the literature 6 planting methods that was commonly used. And I put it on the gradient of the distribution, the perturbation. There is the plantation in the understory of the forest. In the planted trees don't have access to many to the sunlight. The line planting, they open the narrow pathway in the forest. The gap, the degraded area. This one, it's a degraded area that we can have big trees, but the sunlight reach the ground. Then seedling can grow. The growth methods. Here, it's when we prepare the area, with the small seedlines from target species, and we enrich with others that with high value. And in the last, the clear cutting, the highly degraded area that we cut every species, and we plant seedlings from target species for high value. Then we put in our model the guild. Here we have, I told before, a pioneer, non-pioneer, light demanding, and shade-bearer species. We consider also the forest type, the dispersal modes. We had in assist animal and wind dispersed species. We consider wood density and leaf phenology. At the second part of our models, we put the site and the species as a random effect. Why the species in random effect? Because we did not have many observations to compare species each others. What did you find with these models? Considering the tree survival, we found that the mortality is high at the 7 first years. And it's better if we want to put some activities to improve performances of tree species in the plantation. For example, maintenance, to concentrate it at the 7 first years. But we were not able to identify other factors that influencing tree survival with the variable that we considered in our models. Considering the high and diameter growth, we found that in the degraded areas, the regrowth and clear cut, all guilds presented the poor results. As you can, the good result, as you can see here. But in the gap, as you can see here, the gap had the worst result for all species, except the exception it's from high growth and only for pioneer species. This it can be explained by the fact that pioneer species look a lot of quantity of light, and in this environment they are very competitive. Then they grow slowly to reach the to stay in sunlight. That we can explain the high performances of pioneer species. The question remains, at the species level, what can we learn? As you can see, our analysis was done on 89 species, but we didn't have many observations per species, and it was not able for us to inference on species level. But as you can see on this map, as you can see on this map, the variability on tree performances, here it's the diameter increment. The variability is high depending of planting method used. Then it's better to choose the best planting method for each species to have the best performances of trees in the plantation. And if you know the right planting method for a species, it can improve restoration success. Then we try to arrange the tree performances, the tree species according to their performances.
Species according to their survival and the diameter growth. As we said before, high growth is depending on the capacity of a species to fight sunlight. it's related to the competition. We can find that we have some species who show good performances that we call good performing species, but others were poor performing. Here, you can see that the size of the point is related to the mini plantation age, and the color is depending on the species guild. We have here in red pioneer, in a green non-pioneer light demanding, and in blue for shade bearer species. We can note that these results are based on the literature collected in the whole Guinea-Congolese region, which is very high with different environmental conditions that can affect our results. But we must know that it's important to have some procs, some base at which we can learn. We must work to know more at the site level. Then, what are our research perspective? We will verify these literature-based research by modeling the survival, height, and diameter growth of 56 species planted in the gaps. We choose two kinds of gaps, logging gaps and log yard. And already we collected data, and we analyze our data set. And also we will do the same analysis on degraded area. And they will do it in a country, in Cameroon. It's a Central Africa country. But as our preliminary results show that we have some species at the gap that had the good performances. We can see, for example, Terminalia superba, Bionella toxisperma, and Erythrophlehon suaveolense. This picture it's for logging gap. This one it's for log yard, and you can see here an example of degraded area. What can take as a take-home message from this study? The 7 first year, it's very important to improve the planting success. Then we must work hard at the 7 first years by maintaining plantations to improve tree growth. And an appropriate correspondence between species guild and planting method is very important in planting. Then we must choose the species with high survival and diameter growth to improve the success in the restoration initiatives. To have a big tree like this tree like we call Ayus. In the international market. Thank you.
Thank you very much for your talk, and I think that now we can invite our next speaker, James Aluborade, to give us a talk about needle structure and phytochemical efficiency along a needle age gradient in 4 species of evergreen conifers. The floor is yours.
Good afternoon, everyone. My name is James Aluborade. I'm a master student of Poznan University of Life Sciences. This afternoon, I'll be talking on needle structure and photochemical efficiency along a needle age gradient in 3 species of evergreen conifers. I'll start with the background of the study. This study focused on needle photochemistry, and I expanded my research by investigating the relationship between the aged needles and their structure and photochemistry in 3 evergreen conifer species. The species I worked on were Tarsus baccata, Picea albis, and Albis arbor. I tested the following hypothesis. the needle photosynthetic performance, I tested that it's going to decrease along the increasing needle age, and it's going to lead to self-shading in tree crown. That is, the needle, it's going to decrease, the photosynthetic performance of the needle will decrease from the youngest to the oldest, that is from the outer of the branch to the trunk. I also tested that the leaf mass to area ratio would reflect leaf structure, and it's going to non-linearly increase, and along the line it's going to get stabilized at a particular year. And that I also tested the total chlorophyll and carotenoid contents, that they would decrease with increasing needle age. Additionally, I tested the chlorophyll AB ratio, that they would decrease with increasing needle age. As the needles are increasing in age from year 1 to year 2, 3, 4, 5, the chlorophyll AB ratio rather, would decrease, and that would positively influence the capacity of light capture by hold and shaded needles. Now, what are the methods I used? The first thing I did was I measured the light in tree crown. The relative photosynthetic photon flux was calculated using 2 quantum meters, and if this is a quantum meter, that's a quantum meter. So I made use of 2 quantum meters at the same time, myself and my professor. We took the reading of the outside of the crown away from the tree, some meters away from the tree, and then we took readings from the branches at the same time. Then we did the needle mass to area ratio. Here we were able to calculate, to get some needles. We oven dried them at 65 degrees Celsius for a week, and then we were able to scan them with Winsidu scanner. Then we get the dry mass, and then we divided it with the needle area, and that gave us the needle mass to area ratio. Then the measurement of chlorophyll in fluorescence, rather, this was done with Ansatech. I made use of Ansatech to carry this out. Then the modeling of light curves, I was able to make use of Professor Ye's models. Professor Ye is a mathematician, a Chinese mathematician, so he developed some models, so I was able to make use of his models to do my calculations. Then the spectrophotometrical analysis of photosynthetic pigments. With this, we were able to carry out the carotenoids, the chlorophyll A, chlorophyll B ratio, and everything related to the chlorophyll pigment. Then the statistical analysis. This was done by using two-way ANOVA, and then the means were separated using 2-keys test. And these are the abbreviations I will be using in this. ETR mass, that's electron transfer rate, maximum of it. Then the FV/FM, PPF SAT, LMA, MPQ, non-photochemical quenching of fluorescence. Now to the results. My results, the first one I'll be talking about is the PPF. With the use of our quantum meter, we're able to take readings from the outside of the crown of the tree and then within the tree branches, and we're able to we discovered that Abizaba, it's decreased rapidly from the youngest to the lowest. This confirms our hypothesis and also confirms some works that has been done, including one by my professor, Robakowski and Berlin 2017, that it's going to decrease rapidly from the youngest to the oldest. Then for Tarsus, the same thing, it's decreased rapidly from the youngest to the oldest. Unfortunately for Tarsus baccata, we all know the issues with Tarsus baccata. We are unable to get more needle age, so we are only able to get short. So we decided to short needle age minima, so we decided to use just 4. But for PCRBs, it's decreased, but the decrease is not much. It's decreased at a very low space. Now for LMA, we hypothesized that it's going to increase non-linearly, and that was what we got. It's increased non-linearly, and when it got to year 5, it became stabilized. This proves that older needles, we have thicker compositions than younger needles. Then for PPF SAT, the quantum yield of PS2 photochemistry, this decreased non-linearly. For Abizaba, it decreased non-linearly from the youngest to the oldest. But this cannot be said for Tarsus baccata because of the limited number of needle age. We were only able to get 1 to 4, but for others, we were able to use 1 to 8. Now, there's something about this, what this is trying to tell us. This is trying to tell us that with the decrease from 1 to 8, the amount of light that is needed to attain ETR mass will be less. You know, ETR mass is electron transfer rate, the maximum level of it. So the amount of light that will be needed to attain ETR mass will be less. And then this phenomenon is kind of typical to plants. It shows that they have the ability to adapt to the various degree of light that may comes their way, and that they will be able to optimize photochemical, photosynthetic efficiency in response to environmental factors, age-related differences, etc. My PPF SAT decreased. It decreased for the 3 species, linearly, non-linearly. This shows that older needles, they acclimate to self-shading. When they grow older, they begin to like undergo self-shading. Then my MPQ. For my MPQ, they all increased from the youngest to the oldest. The 3, they increased from the youngest to the oldest. And this means that as trees are getting older, they begin to lose more energy. More energy will get lost. MPQ means non-photochemical quotient, so quenching rather. So they begin to lose more energy. And that this MPQ is related to ETR mass. When MPQ is losing more energy, ETR mass must reduce. It must decrease from the youngest to the oldest. And this confirms what we have in the ETR mass. They all decreased. Because NPQ is losing energy, MPQ is increasing from the youngest to the oldest, ETR mass must decrease from the youngest to the oldest. Then I was able to carry out the chlorophyll or pigment analysis. I have my chlorophyll total for milligram per gram, and this, they decreased rather, and when they get to a certain stage, they just suddenly increased. The same thing for PCRBs, but for Abizaba, it's not, there's no significant differences, so I removed it. So this shows that factors like physiological factors, environmental factors, age factors are all affecting the needle age as they increase. The same thing applicable to chlorophyll total. It's increased from the first year till the middle year 5, and then it's suddenly decreased. So all these are the parameter is non-linear, the trend rather non-linear. So what are my conclusions? I was able to conclude that evergreen conifers enhance resource utilization and resilience to environmental stress, and that they ensure long-term growth and development by maintaining multiple age classes of needles. Also, I was able to confirm that as needles are getting older, they become more shade acclimated. The older they get, they grow, the more they carry out self-shading. Also, there's evidence that there's a predictable and continuous decrease in the ability of PS2 to convert light energy and transfer electron effectively. This is possibly due to age-related changes in the physiological processes of the needles. And that's what I've been saying earlier on, that the decreases, the results I've been getting are all related to age-related changes in the physiological processes of the needles. The result also revealed that the structural changes in needles, notably the photosynthetic pigments, are influenced by age and environmental factors. So I'm able to confirm from everything, from all my work, that truly age and environmental factors will affect needle age as they increase from year 1, from the youngest to the oldest. The needle maintenance strategy of evergreen conifers enable them to thrive in many environments. However, it is not clear whether this will help conifers to better cope with climate changes compared with deciduous plants. This means that conifers, they can stay in, how should I put it? They can develop well in different environments. They can be able, even in deserted place, they can save water. They can protect themselves, and they won't dry up. But when we're talking about how they cope with climate change. This, we believe, is a research that needs to be further developed. So we are saying this should be for future research. Thank you for listening.
Thank you for your interesting talk. And now I would like to invite the last speaker of this session, Quadri Agdollah Anibaba, who presents us can functional groups help explain vegetation development in post-mining sites? The floor is yours.
Hello everyone, my name is Quadri Anibaba. I'm a final year student at the Institute of Dendrology, Polish Academy of Sciences. So I'll be presenting my part of my PhD work on the topic, can functional group help explain vegetation development in post-mining sites? And this is the presentation outline. I will give a brief background information about the topic, then discuss about the statement of research problem. Then I'll proceed to the research question, data collection, how I collected the data and how the data were processed. And then discuss about my major findings and the implication of the research. And finally, what is the major take-home message. Ecological succession can help us to understand the important ecological processes that is taking place, most especially in degraded sites, and which are all in the process of reclamation. And species richness is a very important parameter that can be used to assess the success of reclamation process that is taking place in a post-mining site. So when we can expand this process or this understanding of species richness to now look at it with respect to life form and pollination strategy, which all encompasses functional group types. And then, because life forms and pollination strategy is an important component of ecosystem function. And when in post-mining sites, ecological succession proceed from early stage of establishment of plant on spore heaps, and then over the years we see gradual transition in vegetation development until when it gets to late successional stages. So what is the statement of research problem? It is well known that ecosystem transformation is by mining currently occupy about 1% of the global land area, which also tell us that the importance of mining activity on biodiversity globally, if mining occupy 1% of global land area. And since this degradation is taking place across the globe, most times in order to have establishment or reclamation of vegetation on this post-mining site, they use technical or spontaneous establishment of vegetation. And research has proved that spontaneous establishment of vegetation help to support biodiversity in this post-mining site. So therefore, to understand how life forms and pollination strategy develop in the course of succession, which can also help us to give us more understanding about the success of ecological reclamation on post-mining sites. So we decided to ask this research question that which species are the most successful on post-mining sites, and specifically focusing on life forms and pollination strategy. And secondly, we asked what is the relationship between species richness of these functional groups with time since spontaneous establishment of vegetation. And what we mean by time since establishment of vegetation, we are referring to the age of the spore site. So we want to see transition. How is the species richness of these various functional group types? How do they change with age of spore site? So to answer this question, we randomly selected 60 post-mining sites that are based on spore heap age, size, and surrounding land use cover on around the spore heap site. Then we use remote sensing techniques and the K-mean classification algorithm to classify spore heap sites into various classes. And from our result, we have 5 major vegetation clusters, which could be further be grouped into 3, whereby cluster 1, 2, and 4 can be categorized as those vegetation that are at the early stage of successional development, while cluster 5 are at the mid-successional development, whereas cluster 3 are the late successional stage of development. And when we look at typical study area on the Google map, this is how our sample plots were established. So we then created 5 plots in a cross section pattern, whereby we have the center plot, and then we have 4 subplots in the northeast and west and south directions. And in total, we had 400 study plots. So within each plot, we established 3 meters radius where vegetation were sampled, and then we collected vascular plants and their abundance using Londo scale. And this is a typical sampling design of our study plot. This is the central plot, and these are the 4 additional plots. So for this particular research question, we developed a plant life form using the Runker scale, and this Runker scale is based on it is based on the location of the bud. That is how the various plants were categorized. So we have the first one, which is the phanerophyte, which have the bud mostly at the topmost part of the plant, above 50 centimeters, and above the ground. And then next we have the hemiphytes, which have their bud very close to the ground, around 25 centimeters. So for the phanerophyte, we have mostly tree plants and shrubs, while for the hemiphyte, we have mostly small woody shrubs, and they are often found about 25 centimeters above the ground. Then next we have the hemicryptophyte, which have their bud located near to the ground surface. And these are mostly perennial herbs that are found in the temperate region, like the Thorazacum and the DSC. Next, we have the geophytes, which have their bud below the ground surface. Then we have the halophytes, which are mostly found in mesh areas like salt, waterlogged area. Then we have the hydrophytes, which have their bud very like below water surface or water level. Sorry. Then to answer our first research question, like which species are the most successful in post-mining areas in terms of life forms and pollination strategy, we found that hemicryptophytes are the most successful, and which have about 177 species. And then this is followed by the pterophyte, and next is geophyte. Then we have the phanerophyte and the hemiphyte. For pollination strategy, insect pollinated plants are the most successful on post-mining site. Then when we go further to know which species within these particular functional groups are more frequent, we see for the hemiphyte, the Thymus polygoid are the most successful for hemiphyte. Then for geophytes, we see that Cesium avances are the most successful, while for hemicryptophyte, we found that Calamagrostis epigerus are the most successful. Then for phanerophytes, we have the Robus cesius are the most successful. Then for the pterophyte, we have the Erigeron canadensis are the most successful, with higher number of occurrence in plots, sampled plots. Then for pollination strategy, for insect-pollinated plant, the Darcus carota are the most successful. For self-pollinated plant, we have the Erigeron canadensis are the most successful, while the wind-pollinated plant, we have the Calamagrostis epigerus are the most successful. Now to proceed to the second research question in terms of what is the relationship between species richness and of this functional group with time since spontaneous establishment of plot. We found that phanerophytes increase with age since establishment of plot, which logically true, since phanerophytes are mostly trees or shrubs that have their bud above 50 centimeters above the ground. So we expect that as the successional stage develop, there will be more establishment of trees getting towards clamas community. Then for hemiphyte, we have the decrease, although the hemiphyte for the model, the sample size is quite low, since it's the least occurred in terms of success. So we found a low decrease in the richness of hemiphyte with age since establishment of spore E. For the hemicryptophyte, we also found a decrease in species richness as the successional stage proceed. Likewise for pterophyte, we also found a decrease in species richness. Then for geophyte, we also found a decrease. For insect-pollinated plant, we also found a decrease in species richness as the successional stage proceed. And then for self-pollinated plant, we also found a decrease in species richness. For wind-pollinated plants, we also found a decrease in species richness as the successional stage proceed. And what is the major implication of our research? We see that this research has helped us to understand that we can use plant forms and pollination strategy to help improve description and prediction of vegetation process and dynamics that are taking place in succession, which also help us to reduce the complexity of a successional process, because understanding successional process is quite complex. So having the development of such kind of model in terms of life forms and pollination strategy from our findings, which is it has made it much more simplified to understand ecological succession taking place in post-mining areas. It also we also see that the response of a species through succession are to some extent controlled by life history traits. That is, in terms of the plant forms and pollination strategy. As we see from our result that the phanerophyte, the species richness increase, which is logically true, and that of other life forms like pterophyte, which are mostly annual plants, and hemicryptophyte and hemiphyte and geophyte, they all decrease. So to some extent, the results or the research can help us to understand the process of ecological succession on post-mining sites. And then the richness, the high richness of insect-pollinated plant can also suggest a possible interaction taking place with other biodiversity, most especially insect-pollinated plant species interacting with their pollinating agent like butterfly, most especially in the mid-succession. So what are the major take-home from the research? The study life forms and pollination strategy can help us to understand possible trends that are taking place in the process of ecological succession in post-mining sites. And then some trends are not really surprising, like in terms of the increase in phanerophyte and decrease in pterophyte, and which of course have been reported in many studies, most especially in the temperate ecosystems. And then the one that is quite surprising during the course of the research is that of the hemicryptophyte and geophyte, which in previous literature, they didn't really see a clear trend in the pattern. Maybe due to the fact that for our study, we consulted more at the age of the spore heap were much more longer, because the time since established was around 60 years. But most studies, usually their spore heaps are around 10 years. So maybe that is why they didn't really get a clear pattern in terms of hemicryptophyte and geophyte. And from this, I will like to thank the National Science Center for supporting the research under the grant, linking soil substrate, biogeochemical properties, and spontaneous succession on post-mining site, novel ecosystem in the human transformed landscape. Thank you for your time.
Okay, thank you very much for your interesting talk. And now we are starting the most exciting part, because there will be a question for you and actually a discussion. So I really encourage you to ask a question or even to share to share some thoughts about the presentation, the talk you hear now. So when there will be a question, please, that the presenter will ask a question. So.
I have a question to everyone who thinks about it. Actually, when I was listening to your presentations, I started thinking how fast is the evolution in the forests near Chernobyl, because we can expect that the occurrence of mutations is much higher than other places, but and on the other hand, we know that the nature coped with it, and now there is a lot of plants there, and animals as well. But what is the biodiversity and genetic flow in this place compared with non-radiated areas? Thank you.
So a counter question to your question, do you have like some sort of data that you got the idea from about plants and mutations? Because mostly when I heard about Chernobyl, it was about animal mutations. Was there like some sort of actual evidence for it so we can continue this discussion?
I don't have any evidence. I haven't been there. I just expect that if there is high irradiation, it can provoke higher range of mutations.
I don't know anything about bryophytes in Chernobyl, so I'm afraid I won't answer your question. But last year I had a talk from some Ukrainian botanists from Chernobyl surrounding, and they were doing something about Pinus sylvestris. So if you are interested in this topic, maybe it's something to read about. But I don't think if we if we are competent to talk about. I can check it and we can talk later. Okay.
I have a question.
It's on.
Okay. For the first speaker and the third speaker. Yeah. So for the first speaker, of course, your research was related to bryophytes, and then and specifically for the PCA figure you showed, and where the bryophytes for managed area were less dispersed compared to those in under protected areas. And I want to know, okay, fine, is it due to probably species that are planted in these managed areas when compared to those that are in protected areas? And because also from the functional richness result that you also presented, the functional richness of species in bryophyte species in protected areas were significantly higher than that of the managed areas. So probably, I don't know, maybe you will give me a better explanation as maybe it's due to the species that are in these different areas. So then to the third speaker, okay.
Just quickly. Yeah, so first, there's of course a complex answer to this. But first, and I think the most significant, is that in this protected forest, there is a lot more available of different substrata and different microhabitats. In this managed forest, from my observations and my results, there is less of that wood, less of some heterogeneity of ground level, and I don't know if it's also important that some species of trees were planted, because I was working only on this hornbeam oak forest, so there wasn't planted anything from my observations. But yeah, the thing about availability of substrata of in protected forest, there are also more older trees, and dead wood is also at the different stage of decaying. There's also new dead wood and also very, very old. And in my managed forest area, it looks very different. Just less substrata and less microhabitats, and also species, like I show you, were very similar between the squares. So this is the same set of species and nothing more. And in protected, wide range of species, from this very common to very rare, like, I don't know, Buxbaumia viridis or Dicranum virida. In my managed forest, I had some Dicranum virida, but Buxbaumia I didn't. So, if it's okay.
So for the second speaker, sorry, for the third speaker, for your meta-analysis, what is the sample size? I just want to know.
The sample size, it's 657 observations across 89 species. The literature is, okay, 45 studies. Yeah. Okay, it's enough? Okay.
Okay, anyone else want to ask a question or share?
Thank you. So I've got a question for Crispin, my colleague. Maybe I missed the information because I was taking pictures of you. What I retained about the presentation is that logging gaps are the worst habitats for planting those species, except for pioneering species, right? And I wanted to know if you measured those logging gaps, because we know that in Central Africa, we used to use reduced impact logging techniques in order to minimize the impact on environment, and that usually it's become too small for a pioneer population, tree species, to reestablish. So what do you think about it?
Thank you. Effectively, in the context of low impact logging, we are encouraging to make a low, when we are harvesting timber species, we must have a low surface for perturbation. But the problem is when the surface is very low, the light is not reach the ground for a long time. Then many tree species, you can plant tree species, but when the canopy closed, the light does not reach the ground, then many species planted died. But we found that even for pioneer species, their performances, it's low. And then we are asked many questions, as I told, this is a meta-analysis from a whole Guinea-Congolian region. But we can have particularity in observations when we go at the site level. Then the second step will be to analyze the performances according to sites from two logging companies. One it will be from Palisco company and the Alpicam company, which are closed at the southeast Cameroon. And we have a data set from 198 logging gaps, which was enriched from up to 19 years. And the data were collected each year. We have up to 18,000 observations. They will help us to know what to do, what species are better to plant in those kind of sites. And the important to know is that these two companies have two kinds of management. One are FSC certified with and they use the low impact logging, and another previously was not certified. Then they can have the big logging gap. And in our analysis, we try to know at that site which species present the good performances and what can be the reason. And then I told at our preliminary result, we have 3 species that gave good performances. I say Terminalia superba. We have also Bionella toxisperma and Erythrophleum suaveolense. We can learn that enrichment of logging gap, we can do it, but we must think enough before choosing the species to plant, because the result is worse, as we see in the literature review.
Thank you. Anyone else want to? Okay.
To ask one question to Kadri. So this question is not very relevant to your study, but maybe relevant to my study. So do you think any below ground community on mycorrhizae can help plant succession in the post-mining site? Did you find any study or any literature, or what is your opinion about this? Okay, this question is very relevant to my study, but not maybe very relevant to your study. But I am curious about this question because do you think any below ground community or mycorrhizae can help this plant succession in the post-mining site or not? Do you find any study or any literature? Or your opinion?
Okay. For your question, I think yes, it can help in succession in post-mining site because they have been studies that have shown this positive interaction between roots system and mycorrhiza fungi community. And so, yeah, I think it can help in most especially for plant development, and likewise for enriching the soil. Because, you know, in post-mining site, what we have is not at the early stage of establishment. What we have is not really soil material, but rather coal extracted sediment. So yes, yeah, it's yeah. Okay, okay, yeah. So yeah, so at the early stage, so what we have is just sedimentary rock material at the early stage of establishment. So with time, this is when formation of soil will start. So the mycorrhiza community can help in such regard.
Okay, there are more questions to today's speakers, or some comments about this study, because this is also which is worth hearing and sharing, of course. Yeah.
Thank you. It's on your study. Your result can be extrapolated to other sites, mining sites, not only for coal mine.
Yes, the findings from our results can be expanded to other mining areas, and not just only coal mine. In fact, even other degraded areas, because basically what is happening in degraded area in terms of stressful soil condition, stressful stress in terms of temperature and humidity are also applicable in other degraded areas. So the results from our findings can be extrapolated to other degraded areas. And also in terms of when we study, when we look at ecological succession, some of the findings that have been established, both in terms of areas that have primary succession or secondary succession, we are also having similar kind of finding in succession that is taking place in post-mining areas. So there is quite a wide range of applicability of the results.
Okay, thank you. Any more question from the audience? Okay, so I allow myself to ask a question to the James Olabrade, yeah, because you measure it, I don't know, maybe I didn't catch it very well. You measure it in one place or from in a different place? If in one place, do you think that you can expect the same trends along age for this species gathered from different places, I mean environmentally different?
Kima, if I should get you clearly, you want to know if the trees are located in a single location? Yes, the trees are located in a single location. For the Abizaba, the three of them are grouped together, same with Tarsus and same with PCR. So, of course, environment will influence because shade from one of the trees would affect the other trees. So environment will definitely influence. But apart from the environment, what I really looked into is if the age-related, if there are going to be age-related differences, and I was able to determine and to conclude that, of course, environment will influence both their age-related differences.
Thank you very much. Maybe some other question came into your mind? Yeah.
Yes, thank you. I was missing for a while, so hopefully nobody posted this question before. So my question is for Francesco. Based on your knowledge, what type of system, so forestry technique system, would you recommend for close to nature forest management in this region?
Well, obviously every different situation, every different kind of silvicultural treatment has its own specific, let's say, best possible harvesting system. For sure, in this case, considering that there is a consistent amount of slope, substantial slope, more than 30%, there is, however, a good forest, a stable forest road network. I mean, let's say the fixed road network already traced on the maps. I think that cable yarding with small-scale cable yarders, with the line of about 300 meters, is the best solution from the point of view of the environmental impacts, for sure. But to allow to apply cable yarding, there is the need probably to shift and to improve very much the knowledge of the local loggers on these systems, because they know only tractors and animals. So, from the scientific point of view, I recommend cable yarding, but I know that it will take, I don't know, 20 years probably to arrive to this situation.
Thank you.
But if you use the cable, wouldn't the harvesting intensity have to be much higher for it to be economically rentable? I mean, from experience in Slovenia, usually when they use cable, it's quite high intensity of harvest.
Also, in this case, it depends on what you mean with cable yarders. If you mean the Valentini V400, which is a machine with the line of 800 meters, if you have to set up a so big machine, you need to have a strong, let's say, cutting amount. But there are some models of little tower yarders, always of the same Valentini, for example, the same producer, which are based on smaller line, smaller machines, and obviously also maybe you can reshape the thinning only on some specific parts. Let's say instead of doing a selective thinning all around the forest parcel, you can do a thinning from above, located among some specific part of the parcel, in which you can set up the line, and the cost and let's say the time of establishing the line gives the possibility to have let's say also cost-effective implementation. There is much literature on this topic by Professor Raffaele Spinelli on small-scale cable yarding, if you want to, let's say, go back to this topic.
Okay, thank you. Do you have more questions? Okay, I don't want to prolong if you don't have any question or comments, so still I encourage you to discuss, but maybe in a smaller group. You will have enough time during the conference because it lasts a few days. But I would like to thank you very much first to the speaker who gave us very interesting thoughts, and also for the others who ask interesting questions. And thank you very much for the discussion, because as I said, this is the most interesting part, and also give us some future questions and so discuss during all of this conference. Thank you very much.